It's wonderful to be here. We have heard of so many promising mechanisms to actually reduce the treatment burden and enhance bioavailability of all these drugs. So we can start with Barry. Barry, of all these great options that we have heard today, which ones are you more optimistic and excited about?
You mean of the ones we heard today? I have to pick? But there's a lot of interesting stuff, like drug delivery. Again, of course, to state the obvious, we've had this long history now since 2005 of monthly or regular injections of free drugs. So being able to extend that is of great interest to all of us. To do it in a safe method. Again, it's a reminder, though, that the intravitreal injections that we all give every month or every X weeks are very safe, besides being very effective. So what we're looking for are things that can extend that durability while still maintaining those high safety standards. It's exciting. There's so many new things. Again, the Retina, being able to actually get biologics into a drug delivery system, I think that's one of the hardest challenges, because the small molecules, you'll notice that the other agents being looked at, many of them, are the TKIs, which are appealing to us because they're small molecules, they're simpler to handle. They have yet to show their success, but they certainly have great promise. So that is always very exciting, too. So having a new category of drugs, i.e. the TKIs in these extended release, but also having the ability to put a biologic into the eye, the Retina approach is very appealing. So they're all very appealing in that sense. They all serve a need. They all have a basis for existing. So it's an exciting time for drug delivery.
Okay, Bob?
Oh, I can't say much more than Barry said. It's great to have these TKIs coming, hopefully, to the clinic, because they can address both VEGF-A, C, and D, and I really think having a injection period of 6 to 12 months would be such a huge improvement over our current regimen. Of course, we have to get through the FDA, and we don't get credit for extended treatments, and the new guidelines make it a little more challenging, I think, to get something over the finish line, although the companies have figured out ways of doing that. But it is really almost unfair that you don't get credit for the extended duration, and you have to be equal to those anti-VEGFs, which work well and are very safe, but are a burden for our clinic. And as Barry said, I really like the ability to give biologics. There's a whole host of new agents coming, bispecifics, new agents that are really hard to deliver into the eye because of the difficulty with either loading or with destabilizing the proteins when you embed them in some sort of polymer. And apparently Retina, I know that they've been able to show efficacy of Avastin at 6 months in an animal model where it works in that situation. So it's nice to see biologic activity of anti-VEGFs at 6 months. And if we can have that platform available for these newer agents that are addressing a host of diseases, I find that the most exciting, really.
Well, platforms that provide enhanced bioavailability to a host of different molecules and drugs is very appealing. So I have a question for Carlos. Carlos, the PDS. The PDS actually was first launched to be used with Lucentis, but what is the potential for the PDS moving forward as far as a delivery system for different drugs?
Thank you, Maria. I think you're spot on. Really, it's a platform delivery system that we really have here. It's the first molecule that we've been testing it on. It has been ranibizumab, and we've been lucky enough that it's a very stable and novel molecule. However, we intend to actually include other molecules as well, and we're actually on the verge of starting our new venture into using it with a new molecule by the end of this year, hopefully, once we restart implantations with cefivancimig, which is our first DutaFab that blocks Ang2 and VEGF, basically bringing the benefits of faricimab and into the port delivery system and being able to maximize that efficacy over long term.
Okay, and what steps, I know there have been problems, with the PDS, so tell us what steps has the company taken to enhance safety?
That's a great question. I think that there's definitely several different topics that we need to cover here. One is with regards to patient selection. The other one has to do with the surgeon itself. And then lastly, with the device. I'm gonna focus on the surgeon and the device parts as well. So with regards to the surgeon, I think the individual skills of the surgeons vary from one to another. It's definitely not the same for an eye to be operated by Maria Vericol versus an eye being operated by me. And second, there's also a component about the learning curve. So the more surgeries that the surgeon performs, the better it becomes over time, thereby improving the safety profile of the procedure itself. Over time, the technique has been evolving, and I expect it to continue to evolve, to be refined, and improve over time as more and new tools become available, but also as we find more and more evidence from our trials as well. So I expect that to keep improving. And then lastly, in the piece where Genentech has been focusing a lot is on the training, making sure that we approach this in a systematic way as much as possible as pilots are trained to be flying. Through dry, wet labs, virtual reality training, pre-case training discussion, peer-to-peer discussions to ensure that those skills are actually actively and effectively transferred. We also have a team of what we call surgical device liaisons who are actually in the OR with the surgeons, helping ensure that the IFU is followed, but also to try to transfer any knowledge or experience to make sure that the surgeons are actually well equipped to address any potential scenario that comes up during the surgery. And then lastly, I think that when it comes to the devices, I believe most of us in the room are aware, last October we actually voluntarily recalled the implant because of septum dislodgement, which is a device deficiency that we found in our trials. And from that time on, our technical team has done a phenomenal job identifying the issues and implementing solutions to the system, because this is really a system by itself, with different devices. The implant is one of them. But all of the system has been improved, as well as the manufacturing processes. And we've added additional quality measures to ensure the maximal quality of the system is actually provided to the patients. And so far, with the preliminary results that we've seen, they're very promising, and we're expecting that we're going to be restarting implantations by the end of the year.
Thank you, Carlos. And Carlos, what molecules are amenable to be used with the PDS?
I think it's a great question. Again, this is really a platform. However, you can't just put anything on the device itself. Things like size, like stability, especially stability, I think, are so important because you have to take into account that this molecule has to have the ability to be on the device at body temperature for over extended periods of times. We're lucky with ranibizumab, and we've seen now over 3 phase 3 trials in 3 different indications that we are able to actually have that sustained delivery with a certainty of outcomes that we haven't seen with intravitreal injections so far. But that's yet to be proven with other molecules as well. And that's one of the things that has us so excited about this.
Thank you, Carlos. So, Caroline, what is the potential for sustained delivery for complement modulators? What do you see?
Thanks, Maria. Good question. I think we've seen that we've pivoted since 1995. Barry remembers with the ganciclovir implant. Now we're treating older patients, and the treatment burden, whether it's complement inhibitors, and some of those patients also need anti-VEGFs, or they might need other things, it really can't be sustained for a prolonged period unless we work towards the next step. And also, I think with complement inhibitors, we've seen increasing effects over time. So it's very important for these patients to stay in the game. I think that the next step would be to have a slow-release molecule, and that might change the characteristics of some of the drugs. If it's possible, they may not need pegylation if it's possible to do that, because the PEG is what helps to increase the half-life. And then I think we need to look at the next steps, whether that could be gene therapy. There's some promising new technology from Roche looking at the factor B antisense. So I think that we have to do what we've done with anti-VEGFs, find our building blocks, and continue to work on that to the next step. And Maria, like all the talks that we heard, really attest to the imagination and how everyone in the retina space thinks widely, the companies and even the doctors. I mean, everyone adapts and finds the next best thing and tries many multiple things for the varied patients that we have. So it's really amazing.
Thank you, Caroline. So we know that the suprachoroidal space is a promising space, and it's easy. We do it in the office. So for Barry, how do we measure actually concentrations in the retina in these patients? And what are the limitations of the types of molecules that we can actually use in this space?
Well, thanks, Maria. Again, the data is from animal studies, and we saw a little bit, George presented a slide there, but it is interesting that with suprachoroidal delivery, and again, the experience so far has been with two compounds in particular that Clearside has sort of led the way with triamcinolone and also then a TKI, axitinib. There seems to be this significant concentration of drug levels in the retina, again, in animal models, beyond what you see in the vitreous, a 10-fold roughly. He was shown 11-fold, but I think of it as 10-fold X, which is quite remarkable with great durability. In terms of the types of drugs that would be there, certainly we've seen this big divergence between a steroid and a TKI. I don't know that they've explored the possibility of biologics in that space. Again, it's more of a drug delivery route per se. Again, as has been alluded to, stability of drug over a prolonged period of time becomes an issue, though by and large even the biologics can be stable inside the eye for a prolonged period of time, though the Rivanna approach, I should just allude to, again, not suprachoroidal, is a solid system, whereas the Roche Genentech one, the PDS, is a liquid system, so there's different stability characteristics. I would almost ask, Caroline, is the suprachoroidal approach a viable option for something like the complement inhibition, even with the pegylation? I think that's stable and could be placed in the suprachoroidal space. We've also seen gene therapy going in that space. So the opportunities there seem quite broad. It's an area that we're all still learning more about, but that suprachoroidal space is potentially a solution for a longer delivery approach to even the complement inhibition, which we'd prefer to have, again, obviously much longer interval between injections if possible.
Okay, and are water-soluble molecules a possibility to be used in the suprachoroidal space? Bob?
I guess it's a possibility. I would posit that the like the depot, like a triamcinolone depot, is more likely to last a heck of a lot longer than something that's water-soluble. I think it would likely be flushed into the choroidal blood flow pretty quickly. So I would think that longer-acting agents would be more likely to be depots or big molecules, pegylated big molecules, or something of that sort. The TKIs are not particularly soluble, so they do well, in that space. So I would say probably not a good long-term therapeutic. But if it's harboring gene therapy or something, it just needs to be there for a short period of time, it'd be fine.
Okay. So another question. We have at present several drugs that provide sustained effect for up to 4 months and some coming up. So what time is the holy grail for this? Is it a one-and-done gene therapy that will provide us with treatment forever? Is it 6 months? Is it a year? So we can all, everybody can give us their opinion on this one.
I think the route of delivery is important. Gene therapy, if it's subretinal requiring a surgery, as appealing as the one and done, it still is more invasive. If there was an intravitreal injection of a polymer, let's say, that was office-based, that would last, to my mind, the sweet spot between 6 and 12 months, that has appeal too, because we do want to monitor the patients, so the patients are coming in at some interval anyway. So if we had, I mean, I think to me the big debate would be, which would you rather have, let's take the halfway point between 6 and 12, 9-month drug delivery system, intravitreal in the office, versus a surgical subretinal delivery. That would be a one and done, though. That's an interesting debate. I'm not sure how I would end up on that debate. They both sound very appealing. The others have, I'm sure, their opinions.
Caroline?
Well, of course, it depends on what the disease is. I think if we're talking about diabetes, I know, Maria, you'd just rather do a vitrectomy. But
Of course.
so I know how you think. I mean, it depends what the disease is. I think something that's more permanent with a younger patient, I'd actually be a little more concerned with not seeing them for follow-up. But certainly our older patients, the treatment burden on them to come into an office and have multiple treatments is really huge. And I'd like to see something that would last 4 to 6 months. I think that would benefit our patients, and yet we'd still be able to see them. And of course, in saying that, when we have home OCT, we might not even need to see those patients as much.
And Bob?
I tend to agree. I think, I guess the sweet spot is probably 6 months now with our 8 milligram aflibercept, and along with faricimab. I think we expect longer duration between treatments, and I would have answered differently a couple of years ago, and I would have said, 3 or 4 months would be great. But we're sort of getting to that for a large percentage of our patients for a lot of the diseases now with those 2 agents, or one coming. So I think I would push it to 6 months to make it more worthwhile. And I would love to have longer, 9 months, 12 months, and things like Oculeaf may go that far with the huge volume that they can load. But I'm not opposed to injecting, not every time I see the patient. I'd love to have a 12-month interval of injections, but see them as much as I need to in between. And you're right, with a home OCT, maybe don't need to see them at all. But I would love to have the longer the better. But the minimum to me is probably 6 months, given what we have now and coming.
Well, thank you so much to my panel. This has been wonderful. You made my life very easy. So we go with the...
Thanks, Maria.
Yes.